Hey everyone, my name is Joshua Prater. I am currently studying for my PhD in psychology. Even though my presentation has bullet points, much of my information we'll be talking about in my presentation is in my notes section. Please follow along and let's get started. Today, my presentation will focus on traumatic brain injuries or TBIs. I decided to do my presentation on the article Inhibition of 2-AG Hydrolysis Evaluates Post-Traumatic Headache attributed to mild traumatic brain injury. The the research conducted by Wing, Tanaka, and Zhang will be observed and understood to help advance our understanding of this neurocognitive disorder. This original research inhibits 2-AG hydrolysis by utilizing medication to evaluate headaches associated with TBIs. The researchers The researchers used a few different medications and subjects to compare the results and the assisting validity. I decided to discuss this article and its effects on neuropsychology due to my history with TBIs. Unfortunately, I dealt with 4 concussions when I was a child. Several of those I don't remember, and it has affected my short-term memory. While reading this study, I understood from personal experience the effects of PTHs to TBIs. Currently, I still deal with the effects. I have issues with short-term memory. This study can assist people like me by providing more research understanding on how my brain is working and how to assist myself. First, we will discuss the background and the purpose of the research. Wynn et al. In 2024 discussed the effects of 2-AG hydrolysis on post-traumatic headaches attributed to mild TBIs. The researchers believe that 2-AG helped mitigate the symptoms of post-traumatic headaches or PTHs, even though the pain of these headaches does not completely subside. PTH is one of the most common symptoms associated with TBIs, especially with the moderate to severe types of TBIs. PTH emerges from 7 days within the TBI injury. Patients can experience persistent headaches lasting months or even years, and they can resemble migraines even. Enhanced nociceptive signaling in the trigeminal vascular system, or the TGVS, can contribute to the initiation and propagation of PTH like migraines. The trigeminal ganglion TG receives nociceptive inputs projecting to the meninges and cerebral acetylcholine, transmitting these signals to the brain stem and higher brain centers. Medication can be used to assist with pain management for PTHs. However, they are not often efficient pain relief for TBIs due to the complexity of the PTHs, creating an unmet clinical need. The lack of therapeutic agents and the lack of the therapeutic agents for the PTHs. Endocannabinoid systems, or ECSs, have been shown to be effective in treating TBIs and other various types of pain. The ECS is composed of the endocannabinoids such as the 2-acrylcondylyl glycol, or 2-AG, and the anandamide, or AEA, the cannabinoids type 1 and type 2, which are CB1R and CBR2 receptors, and the enzymes for their synthesis and degradation. SES is ideal for treating both acute and chronic pain due to the on-demand synthesis in the key regions of the nociceptive pathways and the avoidance of the psychotropic side. The effects associated with the uh, opioids, patients with migraines have low levels of the AHA. There are limitations in studying the post-traumatic headaches. The weight drop model rarely produces the speed acceleration characteristics of a head trauma. A controlled cartilage will impact and fluid uh, percussion-induced TBI animal models can require open surgery and can cause damage to the dura mater. Utilizing the closed head impact model of the engineered the rotational acceleration or the chimera could avoid these limitations due to the non-invasive surgical methods. Endocannabinoids are used to treat PTHs related to TBI. However, migraines have not been explored yet. These studies found that the treatment with the MAGL inhibitor MJN110 surpassed the TBI-induced pain. The purpose of the study is to observe if the endocannabinoids can be mitigated can mitigate the pain from the PTH is related to TBIs. The researchers indicated that the need for the further research is used of the ECSs for treatment for PTHs and TBIs. The hypotheses are these here. There are clinical and endocannabinoid deficiencies and the pathogenesis of migraines. Um, really want to see the effect of these ESCs and see if we need to use more of them to the help mitigate the pain for TBIs and help cure TBIs and the PTHs related to TBIs. Controlling the 2-AG metabolism might be crucial to the modulation of pain. That kind of piggybacks off the hypothesis before this. Throughout the research, they supported the hypothesis through utilizing the research. Research utilizes the MJN110, CBR1R, CB2R, D034, 10-week-old male mice, the Chimera device, anesthesia, saline, DNA synthesis kit, the QRT-PCR. The subjects are the mice. The researchers put the animals to sleep with anesthesia. The researchers then used the Chimera to create PTHs and TBI symptoms. So those are the materials and how the materials have been used in the research project. Of course, like I said, the subject of the mice, the researchers put the animal to sleep with anesthesia. The researchers then used the Chimera to create the PTHs and the TBI symptoms. The mice are placed in plastic red tube restrainers while being used with the drugs. All the drugs are used to dissolve into the DMSO saline to provide to the subject. After 7 days, the mice are euthanized and their brains were dissected out and currently frozen. The data was analyzed of variance one way ANOVA to compare the data obtained, PCR, masks, skepta matrix, cell number, and protein images among the different experimental groups. So in short, they used a control group of mice that didn't induce with the TBIs, one that did induce with TBIs. They had a cure group, where they showed a cure using the ESC models and the MJN110 medication. They used different type of medication to compare to see if it worked. They use different type of methods to ensure they can measure this MJN110 properly. The result illustrated that MJN110 had no effect on the loss of consciousness caused by direct TBI impact. The results further illustrated that the MJN110 treatment prevented latent pain, hypersensitization in the RN-TBI animals. This was illustrated using the injection of the CC3P at 1.01 milligrams per kilogram on the days 33 and 45 post-TBI. The group showed aggravated allodynia at the lowest threshold of 27.27 grams a day on day 33 and on 31.31 grams on one day on day 45. And 110 suppressed the TBI-induced paraorbital anadola. On day 14 post-TBI, animals treated with the MJ110 had increased the tactical threshold of 1.06 grams, which is higher than the other TBI groups at 0.6 grams. A significant difference was only observed in the MJN 110 and the MA281 treatment groups at 14-day post-TBI, illustrating MJN110 therapeutic effects. Anti-inflammatory effects of the MJN110 at TG and TCNC of TBI mice was reversed by the cannabinoids. Inflammation is caused by PTHs related to the TBIs. First, let's talk about the strengths. In this session, we'll talk about strengths and weaknesses. Let's talk about the strengths first. The experiment was really detailed. The materials used were well thought out. So were the subjects. The researchers tried to cover every detail to achieve valid results as much as humanly possible. The researchers used a lot of terminology that was difficult to understand. However, they were able to explain each detail of the terminology to make sure it was easy as possible to understand. The hypothesis was made clear at the beginning of the study, and they answered the hypothesis, and the information support the hypothesis. The impact of the study can further neuropsychology by finding what can cure and mitigate damage from TBIs. If researchers can get to this point, the understanding of the human brain will increase. The study addressed the strengths and weaknesses it may face and try to utilize the materials that can cover the experiment's weaknesses. The focus of the research is something in need of further research, so it provided relevant information. Next, we'll talk about the weaknesses. Even though the researchers attempted to use a non-invasive way of conducting their research on living beings, the researchers still killed some of the mice and preserved their brains to study. This is called euthanization. This can cause a disagreement in ethics. Mice tend to be one of the main organisms used in experiments. However, conducting experiments that can take any living being's life can be viewed as unethical to some. Even though the injections cause TBIs and PTHS is for the experiment can cause ethical issue as well. When conducting experiments on living beings that are not human, how can we get accurate data on the effects of human beings? There may be similarities in anatomy between living creatures, but there are differences. The research failed to acknowledge that factor. The medication can be successful on mice, however, what effects does this have on human beings? This will take further research, but we need to conduct it ethically. The article failed to explain what the data meant. The researcher explained the impact of the data, but failed to explain what the numerical values meant to the audience. Even though the researchers made it clear the impact data did not give the audience the ability to understand the data on their own unless they determine this for themselves. To conclude this presentation, I would like to discuss the significance of the findings on the field of behavioral neuroscience. These findings are significant to behavioral neuroscience The research broke down details of the differences in proteins and medication to assist the symptoms of TBI. Also, it helps students and other researchers understand TBIs with the chronic traumatic endoseptal for the CTE and becoming more apparent in researchers understanding it more. Researchers such as this can contribute to even more detailed research. This can save a life because there are people who lost their lives to TBIs. This research even goes into possibly long-term effects of TBIs and how medication can assist with recovery. In the study, the researchers mentioned the lack of research on the therapeutic effects of medication on the TBIs. The study provides some research needed. Since the study is focused on mice, how can further research be done ethically in a manner to achieve the research needed? If the cure TBIs can be found, behavioral neuroscience can be find new direction for research. It will indicate a better understanding of the brain. I would like to close this presentation by the importance of emphasizing the present dangers of TBIs can bring. Some TBIs have led to death. There are some news stories of football players passing away after severe head injuries or their lives being altered due to the TBIs. I have had 4 concussions myself, however, I remember only how 2 of them occurred. The other 2 I had to rely on witnesses to tell me how I got the concussions. Hopefully experiences like this can bring sense of security to those who need it. Thank you for your time and listening to my presentation. Okay.